Hello, students, and welcome to my photosynthesis screencast for AP Biology. Just like all my videos, this isn't for profit. This is just meant to help you. Here, I hope that you'll appreciate the soccer pitch and trees and parks you might go to as you're thinking about this, and that we couldn't be around without photosynthesis, right? Photosynthesis evolved first out of photosynthesis and cellular respiration. It put oxygen into the air, and it gives us the ability to break down these carbohydrates. It's really beautiful, and it's something to think about as we're trying to take better care of our planet and do better environmental policies. Okay, check your knowledge. Can you do these? This is what I'd like for you to get out of it. I felt that you were doing a great job in class. I was pleased with your reading quiz responses on this. You did a cool modeling of the Calvin cycle with Plato. And then when I asked you the lecture questions in class about the light-dependent reactions, I thought like you did well with them. And then I gave you practice multiple choices and some free responses, and I thought you really showed your knowledge. Okay, so here's a 9th grade equation for it. Real simple, right? Carbon dioxide, water, and light coming in and making carbohydrates and oxygen. It's not that simple, right? There's a lot of stuff going on. But that's what we teach in 9th grade, and even less maybe, or we teach in middle school, but now you can really dive into it. So what does it mean to be a plant? It's much more than just doing photosynthesis. Sometimes students get lost. They got to think about that big picture, right? And so they're going to need to get building blocks from the environment. They're going to need to get vitamins and minerals, and they need to get different elements. And so this is pretty cool, right? It's going to show that there can be limiting factors. So when you have fertilizers, they can have nitrogen and phosphorus in them, and those can often be limiting factors. And so when they leach into or get put into lakes or oceans, they can cause those algae blooms. All right, and plants are also going to need, they do more than just making carbohydrates, right? They have to make their own proteins, lipids, and nucleic acids. And I thought you could see that in action with, with the AP free response question that we did about the seeds that were in the fires. I thought it was really cool. Okay. Big thanks to Ms. Foglia for that slide. All right, so it's going to be the light reaction, which is going to make ATP and NADPH, and then the light independent reactions, which are going to use ATP and NADPH to fix carbon dioxide to ribose biphosphate, and then it's going to reduce that into an energy-rich molecule that is the precursor for glucose. And then it'll regenerate the RuBP. So let's go through it. All right. CO2 on its own has very little chemical energy, but when it's reduced, when it's highly reduced in the form of glucose, it's going to get a lot. And right, so that would be the reduction part, but you can never have reduction without oxidation. They go together. And what's going to be oxidized is the water into oxygen. So if you look at the equation, you can see that. This is an anorganic process. So what does that mean for delta G? Think about that. It's going to be a positive value, and you can go through some examples of that. Where is that energy coming from? It's coming from photons of light from the sun, or maybe from like a hydrothermal vent, right? It's not always plants. There can be things like cyanobacteria, even some protists, et cetera, that are photosynthetic as well. And this preceded cellular respiration in evolutionary history, right? Because it had to make oxygen and make sugars to be broken down. So it came about early. All right. It takes a lot of steps, right? You can't just go from this energy poor molecule to energy rich one. It's going to have a lot of different enzymes that play there, and it's really amazing, right? And so some of this GMO stuff is looking at ways that maybe some of those enzymes or other pieces can be altered. Okay, so it occurs in the chloroplast. You should know what these are, and then these little green pancakes are going to be the thylakoids, and within them you're going to see a lot of action happening on the membranes. And from cellular respiration, you should remember that like the membranes are going to be they're phospholipids, so they're going to be able to separate charges, and they're going to make that chemoosmotic gradient with protons. This is just a little terminology review. And then A lot of you answered this on your quiz, dealing with the different pigments. We will finish this. I had to get the chemicals for scratching it on paper and seeing the pigment separate. And so with chromatography, and we'll do that as well. So you knew about chlorophyll, and that's why plants are green, right? They don't absorb green very well, so they reflect it. And then there's also carotenoids and some xanthophylls, and they absorb other spectrum colors. And then these also help to dissipate excess heat energy. So visible light, it's not just white, right? It's got a lot of different colors in it, and it gives you what you see. So I tried to put some fun pictures in here of what light might be bouncing off. And this is just my boy when he was little. Okay, so we went over chlorophyll. I'm going to speed up a little bit. Remember that these are all on Google Classroom. So I do want you to remember one thing you definitely went over in intro bio is that photosynthesis and cellular respiration are intricately linked, right? The products of one are the reactants of another. And so if one were to stop, then they both would come to a halt. And so they're both very important. One question for us is, are we putting tons of carbon dioxide in the air in addition to what would come out from normal cellular respiration? Okay, quick student misconception, right, that plants wouldn't need to undergo cellular respiration, but they do, because they need to make ATP through cellular respiration. They can make, or they do make ATP during the light-dependent reactions, and they have something called cyclic photophosphorylation, which can keep making ATP, but they're going to need mitochondria as well. Here's that redox equation that I was talking to you about. So now you can see the whole thing. So CO2 is being reduced to glucose, and water is being oxidized to oxygen. They always go together. And you can remember oil rig. Oxidation is losing electrons. So these electrons from water are going to be stripped, and they're going to go through the electron transport chain. Reduction is gaining. So here, CO2 is gaining those electrons. And where are they getting them from? They're getting them from NADPH, which I told you to think about as kind of like a taxi cab carrying electrons and hydrogen protons. This is a nice model that shows the interconnectedness between the light reactions and the Calvin cycle, or the light independent reactions. And you should definitively be able to describe this model right here. You should also know that this is a model, right? There's limitations to it. It's not like this happens on the left and this happens on the right in a chloroplast. Right, there's lots of thylakoids around, and then there's the stroma, or the fluid with the Calvin cycle enzymes in it. Okay, when you think about the light reaction, you should be thinking about three things. You should be thinking about photoexcitation. Basically, that means that the chlorophyll molecule absorbs some of that light energy and it loses an electron. That electron then goes through the ETC, or the electron transport chain. As it goes through, it loses free energy. That free energy is used to pump protons, and those protons are going to congregate and then go through ATP synthase. This is pretty cool. It's the opposite of cellular respiration. In cellular respiration, we pump the protons out, and so they're in the intermembrane space, but in photosynthesis, they're pumped into the interior of the thylakoid. And in chemiosmosis, remember the word osmosis, diffusion, right? So these chemicals, or the protons, are going to diffuse through ATP synthase. All right, there's two photosystems. It's a little complicated. Photosystem 1 is after photosystem 2, but it's found first. And I don't think you're going to need to know cyclic and non-cyclic photophosphorylation, but we'll go over it anyway. So here, Photosystem 2, let's just say that light hits it. So it's nice and sunny outside in Oman. The electron is going to be passed through the chain. Well, the loss of electrons here, they need to be replaced, right? Oil rig, oxidation is losing, something needs to be reduced. And so where are we going to get those electrons from? We're going to get them from water. So we're going to split water. We're going to oxidize that water from H2O into oxygen. What is getting reduced right here? What's gaining them? Well, NADPH. And so water is split. The oxygens grab onto another one. I'm sorry. I don't know why this thing keeps popping up. Let me go here and go back. Okay. So this half oxygen down here, it was going to combine with another one and make an O2. The H's are here inside the thylakoid space, and they'll eventually diffuse through ATP synthase. This is called non-cyclic or linear electron flow. At Photosystem II is where this started off, and so this just tells a little bit about the chlorophyll molecule and the enzymes involved with it. And then here's just an example of the water being split. So this is a cool experiment, right? So if you put some tracers on that oxygen, you can see, well, where does the oxygen in the air come from? Does it come from the water or carbon dioxide? And you can see that it comes from the water, right? So the tracer was put on the water molecule, and you can see it coming out as oxygen. If you put the tracer on the carbon dioxide molecule, you can see it coming out in glucose. Okay, so let's keep going. Now we're going to say that, well, this is just an overview of it. Once they get from Photosystem II to Photosystem I, they need another bump in energy. And so they're going to get another photon of light bumping them up, and then they're going to eventually go through some enzymes and get into the taxicab from NADP to NADPH. And that's what this was showing. Okay, so now it's excited electrons pass the enzyme NADP plus reductase, and it's going to become NADPH. So that is non-cyclic or linear electron flow. Let's say that the NADP pluses, that there's not enough taxicabs, just like in fermentation in cellular respiration. Well, what's going to happen? Instead of shutting down, it can send the electrons back and keep on making ATP. All right, the chemiosmosis just means that the protons flow through ATP synthase, and it's going to make ATP. Here's an overview of it. All right, so it's a one-way street. Here's just showing stomata. I'm not sure if we did that in class, but we'll need to. So we'll take some fingernail polish and put it on there, and we can make sure that we can see them. You can do some cool little experiments, right, about which plants have higher density of stomata and what are some of the evolutionary benefits of opening and closing them and other ways to fix carbon dioxide, C3, C4 plants. But I'm getting a little distracted. Okay, so that was non-cyclic or linear. Cyclic photophosphorylation is, all right, NADPH is full, send it on back. And so we're going to send it back to Photosystem II, and we can keep using that to make more ATP, right? This is interesting. It's good, right? It requires more ATP than NADPHs to do the Calvin cycle. So it's a way to get more of them. All right. But you would still need light to have this going on. Okay. So, thank you, Ms. Phobia, for this slide. Hopefully that made the light-dependent reactions clear. The Calvin cycle is going to be a little easier for us. It's very complicated in and of itself, but AP has generously said we just need to know 3 things. We need to know fixation, reduction, and regeneration. And you all did the TED Ed video with me where we kind of went through and made all the balls of carbons and saw how they get rearranged. I thought it was pretty fun. So there's this enzyme Rubisco that's going to fix carbon dioxide to this RuBP molecule, ribose bisphosphate. Rubisco's arguably the most important enzyme in the world, and it's maybe the most present enzyme in the world, right? Because this is going on. So once it fixes that carbon dioxide onto then the plant's going to use ATP and NADPH to reduce it so that 6 carbon molecule splits into 3s, and it's going to gain all those electrons and hydrogens, and it's going to become an energy-rich product. And one of those is going to leave. It can go off and it can become sugars. It can be a precursor to be put into proteins. And then 5 of them go this way because you got to remake the starting block. You got to remake the RuBP. And so we had a little fun doing that in class. So all you need to know is fixation, reduction, and regeneration. And it's not this Calvin cycle. I used to love me some Calvin and Hobbes. I had one of my best friends still loves it. It's great. Okay, and so here's just another example of it. Here, I put it in there just so that you would see that and not get worried. This is from the AP curriculum guide, and it says memorization of the steps in the Calvin cycle, names of enzymes are beyond the scope of the course in the AP exam. So don't memorize that stuff. All right, don't memorize stuff anyway. Try and put it all in context and learn through phenomena. All right, so we went over this stuff with Rubisco. You can check it out on Classroom, reduction, and regeneration. Okay, so this was just, I put this in here to show that G3P doesn't just have to go into glucose. It can be the start of making lipids, amino acids, and nucleic acids. Very important. All right, so it's always good to do a summary, right? So the light reactions, what goes in and what comes out. So what goes in, ADP, what comes out, ATP, what goes in, NADP plus, and then what comes out, NADPH. Water goes in, what comes out, oxygen. All right, let's go to the Calvin cycle. What goes in, carbon dioxide. What's made out, G3P, or what's going to become glucose. What goes in, ATP. What comes out, ADP. What goes in, NADPH. What comes out, NADP plus. Okay, sorry. I shouldn't have just read that slide to you. It's getting late, and it's time to go on DO. All right, you should check out the Bozeman video for C3 and C4 and CAM plants. I just couldn't put that in here. It would have been too long. And then hopefully in class, we'll get to go over Rubisco hacking and a case study I've done before on glycine betaine. All right. Like I said, I think you're doing well in photosynthesis. Let's keep rocking and rolling. Check out that free response question that I put in your study guide. And that's an excellent example of what you should know on an AP level about photosynthesis. Take care.